last slammer is not a Martin anymore. We will have Erica. Let me just pull up exactly. Erica Wynne-Jones, who originally comes from Australia, and right now she's a postdoctoral fellow at the National Institute of Health in Maryland, and she will talk to us today about the miraculous ways of our immune system. Erica, the stage is yours.
Right. So I don't know about you, but I have definitely been thinking a lot about my immune system lately. It kind of amazes me how we're doing so much to stop being infected by one virus when every day our immune system is protecting us against trillions of pathogens that we might encounter. So it makes me think about how amazing it is that the immune system does it, and it can do it because the immune system is really diverse. It's made up of a lot of different types of cells that all have different functions. But for me, really, T cells rule, and that's because I focus on viral infections in the skin, and that's why T cells are really important. So I get to work with a lot of cool viruses, including vaccinia virus, which is the virus for the vaccine for smallpox, and herpes, which is herpes. So if we think about the skin, basically its primary function is to be this barrier to the world. And underneath you have lots of blood vessels that transport things into the skin. So when a virus comes along, it often enters through some kind of small breach in the skin, and then it can infect skin cells. So we have a lot of virus-infected cells, and then the immune system sends along these T cells, and they enter into the skin, and then they surround the outside of where the virus-infected cells are, and they all get up close to each other, and they bump into each other. And it really got us thinking about something that you've probably been thinking about too recently, and that is communication. So we really wanted to know, do T cells communicate with other T cells when they're in the skin? And then we compared that to when they're in the bloodstream, when they're a bit further apart. So T cells communicate with these receptors. So these are on the surface of a cell, and basically what they do is receive some kind of signal. And that signal is called a ligand. So these ligands can be on the surface of another cell, so that would require quite close contact between two cells, but they can also be secreted. So you can kind of think of this, say the receptor could be something like an email inbox and the ligand could be an email. So just like our communication, there are a lot of different types of communications that T cells can have with other cells. And mostly these have been characterized by looking at, yes, T cells communicating with other cells, but then we wanted to know whether the T cells can communicate with each other using these receptors and ligands. So what we did was had a look at these T cells in the skin. So do they have receptors? Do they have ligands? And then do they have any pairs so that they can communicate with each other? And what we found was pretty cool. The cells in the skin have 17 more receptor ligand pairs than the cells in the blood. So that's 17 more ways that they can communicate. And what does that mean, really? So there's very little communication between the cells in the bloodstream, but extensive communication between all of the cells in the skin. So it's kind of like the T cells in the bloodstream are social distancing. So even if they did come across another T cell, they're really not going to like shake their hand or anything like that. There's little communication. But T cells in the skin, they aren't just together like on public transport, not communicating. They're really just having this big party. And why is that important? Well, it's really about what the T cells are saying to each other. So broadly, you can have these different types of signals, something that would boost the immune response and combat the virus. So like a go signal, maybe inviting more T cells in or causing lots of inflammation. But that's also balanced by stop signals where the attack on the virus is kind of slowed down, and maybe some T cells, you would get rid of them, and inflammation would be reduced. So all of these extra communications that we see in the skin allow the T cells to act as one big unit as opposed to making individual decisions, so that they can really have a good party. So they're really in balance, there's not too much inflammation, and they're keeping the virus in check. But what happens when this gets out of control is that you can have some excessive inflammation. So if you don't have if you have too many go signals and not enough stop signals, you can have all of this inflammation and cause damage to the tissue. And you do get good control of the virus, but it's at the risk of the other cells. So this is kind of like a bad party. Alternatively, you can also have a sad party. And so that would be like if there are too many stop signals and it's kind of dampened. There's not enough T cells around. So this is also not good, obviously, because the virus is going to spread. But there are some circumstances in medicine where we might want to cause these kinds of, like a bad party or a sad party. So an example would be in cancer, you would want to cause a bad party because all of these, you want to get rid of the tumor, and that's the most important thing. And so the tissue damage just has to happen as a result of that. So there's lots of cancer therapies that target that. And in the same vein, you might want to have a sad party if in autoimmunity, because that's where immune cells attack the body. And so we might want to dampen that. So basically understanding all of these interactions can allow us to potentially have some really positive impacts in medicine. So I'd like to thank everyone, including so Heather, my supervisor, and Tova, our collaborator. And thank you for watching, and may your T cells party hard, but not too hard.